Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about epithelial tissue. Epithelial tissues tend to form sheets of cells that are connected together by a variety of means that we will talk about soon, and these sheets of cells tend to either serve as protective barriers in the body that separate body cavities from each other, separate fluid compartments from each other, and or facilitate absorption and transport processes, which we have talked about before. So to this end, there are two major types of cells that we can find in epithelial tissue. Number one, you have the true epithelia. These are cells that face towards an outside environment, like the skin cells, like lung cells that face towards basically the air that we're breathing in. And then you have what are called endothelial cells. These are very similar, except the difference here is that these cells face towards an inside environment, like the interior of an organ, the interior of a blood vessel, things of that nature. But otherwise, their function and their structure tend to be very, very similar. Let's introduce some epithelial cell terminology. First, we need to understand that epithelial cells are polarized. To this end, polarization basically means that the cell has two distinct ends, very much like a magnet has a north pole and a south pole, and these two ends of the cell are very distinct and have very different functions, largely due to the fact that the two ends of the cells have different types of proteins that are embedded in the two membranes. The two membranes are, number one, the apical membrane. The apical membrane faces towards the lumen or hollow portion of an organ or blood vessel. So if you're talking about the small intestine, the lumen would consist of the hollow space where digested food materials move through. The lumen of the bladder would contain urine. The lumen of a blood vessel would contain blood, and so on and so forth. So the apical membrane, in a lot of cases, is also going to contain microvilli, little finger-like projections from the apical membrane that really serve no other purpose other than to greatly increase the surface area for absorption. And in a previous video, when we observed Fick's law of diffusion, we saw that surface area plays a big role in determining the rate of diffusion and the efficiency of diffusion. On the other end of the cell, you have the basolateral membrane. This faces the opposite direction. It faces towards the extracellular matrix and the blood supply. So another very important thing that the basolateral membrane is going to contain are the sodium-potassium pumps that we have observed before in our video on active transport. And also the basolateral membrane is going to contain a lot of adhesive junction proteins that are embedded in the basolateral membrane and attached to proteins in the extracellular matrix to give these cells some strength and integrity so that they cannot be pulled away from the rest of the protective sheet. Now, one thing we need to make sure we understand about epithelia is that in a lot of cases, epithelia serve as barriers. They serve as protective sheets to make sure that no very large materials pass from one side of the epithelial layer to the other. So one kind of absorptive process that we did not talk about before that can sometimes occur is called paracellular transport. This would occur where a usually very large structure, very large particle or molecule or whatever, instead of passing through membranes, it passes through the empty space in between neighboring cells. So there will be some times when paracellular transport is allowed, such as with capillaries in which the endothelia contain those little gaps in between the fenestrations, but in a lot of cases, paracellular transport cannot be allowed. So a good way to think about this is if you think about your intestinal epithelia, imagine very large materials like the bacteria in the food that you eat. And make no mistake, the food that you eat does contain bacteria, usually harmless bacteria, but bacteria nonetheless. Imagine bacteria getting through these gaps in between the epithelial cells that make up your intestinal lining and getting into the bloodstream. If that's the case, these bacteria are no longer going to be considered harmless, and they will be able to exert harm by getting into the bloodstream and getting into your body. So what helps to protect against paracellular transport in most cases are tight junctions. Tight junctions are a type of cell-to-cell junction that basically seal the edges of neighboring epithelial cells together, making sure that there is no space for bacteria and other large materials to pass through epithelial barriers by paracellular transport. So what this is going to mean is that for something like the intestinal epithelia, if we are going to absorb things like glucose and fatty acids and amino acids and things of that nature, which we obviously need to do, those materials are only going to get absorbed into the bloodstream if we have the capability of getting them to pass through the apical membrane and then through the basolateral membrane. And we obviously had a whole video dedicated to the absorption of glucose in the intestine several videos ago. So if that still seems kind of foreign to you, you may want to go back and watch that. So here is an illustration of several of the different types of cell-to-cell junctions that we will encounter this semester. So the tight junctions that we just discussed are listed here. Tight junctions are composed of proteins called claudins and occludins. That's not super important for a physiology class, but these are multi-subunit proteins made of those two different types of claudin and occludin proteins. And basically what they do is they act kind of like staples that seal the membranes of two neighboring epithelial cells together so that no materials can pass in between the cells by paracellular transport. Obviously, we have seen gap junctions before. Gap junctions will become much more important when we start talking about the heart and the cardiovascular system in the future. And here's some examples of anchoring and adhesive junctions that are going to help anchor those cells down and anchor cells together. So whatever kind of junction protein that you're talking about, for epithelial cells, the major function of these junction proteins is usually either going to be to ensure the integrity of the barrier or to anchor the cells down to the larger tissue, usually by grabbing onto the underlying basement membrane and extracellular matrix. Okay, let's talk about classifying epithelia. Epithelial cells can be categorized based on two different things. Number one, the shape of the cell, which is just a ratio of that cell's width to its length, basically its dimensions. And then number two, the number of cell layers that you find in the actual sheet of epithelia. So first we have the squamous cells. Squamous cells can either be found as a simple layer of squamous cells, which consists of just one layer, or a stratified layer in which you have many layers of epithelial cells stacked on top of each other. And this is going to be the case for all types of epithelia. You either find it as a simple layer or a stratified layer. So what you'll notice about these squamous cells is that they tend to be kind of flattened out like pancakes. They are much wider than they are tall. And as we will see very soon, that really makes them ideal for membrane transport processes, especially the simple cell layer. If we move on to cuboidal cells are cube-shaped. That's probably why they're called cuboidal cells, because they have cube in the name. And they are going to have a fairly similar length and width dimension. So again, they look very much like cubes. And then you have the columnar cells, which are taller than they are wide, hence why they have the word column in their name. In all three cases, of course, you can see what I was saying before. We can find them as simple cell layers or as stratified cell layers in which there are many layers stacked on top of each other. So these are major types of epithelial cells, but what are they good for? So there are two basic things I want to make sure we get across here. Number one, let's imagine the tissue of the lungs. We haven't really talked about the respiratory system yet, but I think you'll be able to grasp this. So in the lungs, we need to be able to transport oxygen and carbon dioxide quickly and efficiently across the pulmonary epithelia. So that's the very thin membranes that make up the alveolar tissue of the lungs. So when we breathe in, we need that gas exchange between what's inside the lungs and the bloodstream to happen as quickly as humanly possible. So the question then becomes, would we prefer for these epithelia to be simple or stratified? Would we prefer them to be squamous, cuboidal, or columnar? So let's take a look at that. So this is an old slide that I showed you before. This was us looking at simple diffusion, specifically in the pulmonary capillaries of the lungs. So what you're seeing here are the squamous, the simple squamous epithelia that make up the tissue of the lungs. So these squamous cells are called type 1 alveolar cells. And the idea here is that when we inhale air, we inflate these alveoli so that the alveoli fill up with oxygen-rich air. And then you can see that oxygen and carbon dioxide are able to move across these epithelial cells by simple diffusion. So, getting back to the question we asked on the previous slide, would we prefer for these type 1 alveolar cells to be simple or stratified? Squamous, cuboidal, or columnar? Well, the idea here is that when oxygen moves across these epithelial cell layers by simple diffusion, it has to cross one membrane, two membrane, and then for the endothelial cells that make up the capillary wall, 3 membrane, 4 membrane. So for the first issue, would we prefer these cells to be simple or stratified? Well, the issue is that if this were stratified, then that would increase the number of membranes that oxygen and carbon dioxide would have to cross. They already have to cross 1, 2, the apical and the basolateral, then basolateral, and then apical here. So that's 4 membranes that they have to cross. If we turn this simple squamous epithelia into stratified squamous epithelia, then they're not crossing 4 membranes. They're crossing either 6, 8, 10, 12, or you get the picture, right? So when it comes to membrane transport processes, whether it's the gas exchange here or the absorption in the epithelia, or in the intestinal epithelia, you would much rather have a simple cell layer than a stratified cell layer because that is going to make the transport go much more quickly. Stratified cell layers are going to be much better served for just serving as barriers, like what you find in the skin. Okay, and then the next question is, now that we've settled on, we probably, for the sake of transport like this, we need it to be a simple cell layer. Do we want it to be squamous, cuboidal, or columnar? Well, so this one's not quite as simple, so you will find some absorptive layers of epithelia, some transport layers of epithelia that will either be squamous, cuboidal, or columnar, but in this particular case, we see that it is squamous. And this acts to our benefit because since these cells are so flattened out, the oxygen and carbon dioxide do not have to diffuse as far because not only of the increased surface area, but because they do not have to move as far in a distance sense. So going back to Fick's law of diffusion, which we saw several videos ago, cell thickness was one of the determinants of the efficiency and rate of transport that we saw before. So if this were a columnar cell and this cell were a lot taller, the oxygen would have to move much further to get from the inside of the alveoli into the bloodstream. But again, that does not mean every absorptive layer of epithelia, not every transport epithelia is necessarily going to be squamous. But in this case, it is a good thing that it is squamous. Okay, so we've talked about membrane transport for epithelia. Let's talk about secretion next. So there are many specialized epithelial cells which are usually either going to function in an exocrine sense or an endocrine sense, and we will define those terms better in the future. These are cells that either produce a protein product or a lipid product that need to be exported out of the cell. That export is called secretion. So exocrine cells secrete their products into a duct or onto some surface, like when you sweat, your sweat glands are exocrine cells and they are secreting that sweat onto the surface of your skin. The exocrine cells of your pancreas produce digestive enzymes, which are proteins, and those enzymes get secreted into pancreatic ducts, which are going to be diverted into the small intestine. So these are protein or lipid products that are synthesized by specialized epithelial cells inside the cell, but those products, whatever they may be, need to be exported out of the cell. So let's take a little look at how that looks. So let's operate under the assumption that this export is going to be a protein of some variety. In this case, protein synthesis will proceed just as we've seen many times before. You have a gene in the DNA that provides the instruction on how to make that protein. Through transcription, RNA polymerase makes an mRNA copy of those instructions, which are exported out of the nucleus. That mRNA binds to a ribosome. The ribosome looks for the start codon, it starts building the protein is released after a stop codon is encountered, the protein folds. But what happens next? How do we take that protein that we just got done making, and how do we export it out of the cell? So after folding, the protein is packaged into a transport vesicle, and for this is usually going to apply to ribosomes that are building proteins in the rough endoplasmic reticulum, because since there's ER membrane already there, it's usually not hard to package that protein up in some membrane that is there. So that vesicle, which you can see here, this transport vesicle, which contains our product, either a protein that was made in the rough ER or a lipid that was made in the smooth ER, that transport vesicle will go towards the cis face of the Golgi apparatus, this big stack of membranes you see right here. In the Golgi, the protein is going to be tagged, usually with carbohydrate chains, and that basically acts as something like a zip code that will help to determine its destination. Some products will stay inside the cell, while other products will be tagged for export out of the cell. So now we have a secretory vesicle which contains our modified and tagged product. That secretory vesicle buds off of the trans-Golgi face and starts moving towards its destination. And in this case, we are talking about the apical membrane up here. That vesicle will fuse with the apical membrane, and that causes the contained within product to be released into either the lumen or into some kind of extracellular fluid. Okay, so as far as secretion goes, we will see more examples of that in the future, especially when we start talking about endocrine cells. But for now, that's going to do it for this video. Here is a list of vocabulary terms you probably ought to be aware of. And for checking your understanding, number 1, what do we mean when we say epithelial cells are polarized? Number 2, what is paracellular transport and how is it prevented in barrier epithelia? And number 3, what are the 3 kinds of epithelial cells and what are they generally used for? Alright, that's going to do it for this video. If you have any questions, go ahead and drop them in the comments section, and I will see you next time. So long.